Hello, I'm Jensen, and I'm currently studying the effects of ethylene glycol on the alpha1 subtype of glycine receptors in Xenopus Laevis Oocytes, and I'm doing so in Dr. Heron's lab. The purpose of my experiment is to determine whether this modulation of glycine receptor function occurs in a concentration-dependent manner. And before I get into this too much, I'll give you a brief primer on what I'm working with here. Ethylene glycol is a component of fracking solution, therefore causing it to become a widespread environmental contaminant, but we don't know the actual implications of this widespread environmental contamination. Additionally, when ingested, it functions as a central nervous system depressant, but we don't know the actual mechanisms of this central nervous system depression. Because of this, I'm studying its effects on glycine receptors, because when glycine binds to the two binding domains on glycine receptors, it causes a conformational change in that protein, which opens up the ion channel pore and allows chloride influx into that cell, which brings a bunch of negative charges with it and causes the cell to be less likely to generate action potentials, which can contribute to central nervous system depression. So in order to do this, I'm using Xenopus Laevis Oocytes as they are a preferred model system for the study of receptor physiology, mainly due to their size and membrane durability. Now that you know a little bit more about what's going on here, my hypothesis is essentially that chloride influx through the ion channel pore will increase in a concentration-dependent manner upon exposure to ethylene glycol. So why I propose this is essentially because ethylene glycol and ethanol have a somewhat similar chemical structure, and I believe this will therefore cause ethylene glycol to bind to the same domain on glycine receptors as does ethanol. And ethanol is known to allosterically increase glycine receptor response when it binds to this domain. So to talk a little bit about the microinjection process, I essentially fill up a borosilicate glass pipette, like the one you can see here, with my RNA sample, and I very controlledly inject 0.05 microliters of gliar alpha1 RNA into each Xenopus Laevis oocyte. I do so in such a controlled manner because the amount of RNA I inject into my cells will theoretically be proportionate to the amount of glycine receptors expressed on the membrane, assuming there is no variable degradation of my RNA. Once I've injected my oocytes, I then allow them to incubate for 1 to 3 days. This just allows these cells to take this protein and express it on their membrane. A brief note about perfusion before we talk about the actual recording process. So first, ND96 solution is perfused over the cells to get a good recordings of baseline cellular function, and then a standard concentration of 100 microliters of glycine is added to get a baseline recording for glycine receptor function. After that, solutions of ethylene glycol are tested to determine how ethylene glycol modulates glycine receptor function. Once these proteins are expressed on the membrane and the perfusion system is set up, I then load my cell into this 2 electrode voltage clamp apparatus you can see here. And I impale the cell with a voltage electrode as seen on the left and a current electrode as seen on the right, both of which are filled with a solution of potassium chloride. I then use this current electrode to inject a steady stream of current into the cell, therefore maintaining the cell's voltage at a fixed value. And by clamping this voltage, I'm then able to record the actual ion currents which are flowing across the membrane through glycine receptors and other ion channels. This process allows me to watch how glycine receptor ion channels respond to different solutions, including ethylene glycol, in real time. If my hypothesis is supported, I expect to see that glycine receptor function is increased by ethylene glycol. When oocytes are perfused with a basic salt solution, they form a baseline level of current flowing across their membrane, as is shown by the 0 level on the y-axis here. Upon exposure to 100 micromolars of glycine, a sharp increase in inward current is seen. This inward current will return back to baseline when the cells are no longer exposed to glycine and glycine molecules are no longer bound to the receptor. When these cells are exposed to the same concentration of glycine, but with the addition of ethylene glycol, I expect to see an increase to negative inward current as the function of glycine receptors is potentiated. I then expect this potentiation to increase proportionately to the amount of ethylene glycol. Finally, as you can see by the rightmost graph here, I expect to see that glycine receptor response is potentiated past its natural baseline when exposed to ethylene glycol. Here are my references. And so finally, I would like to give a big thanks to Dr. Heron for her continuous support and mentorship throughout this project. And I would also like to give a big thanks to the Oscar program for providing me funding for this project in the form of a URSP grant.